right. Great, everyone. So good morning. So today we're going to have a guest lecturer from St. Jude's who will be providing us an overview of the research expertise that he has. So I want to give you a little bit of context of his upbringing. So he received his BS in biochemical sciences at Harvard, and then went on to do a PhD at the University of California in San Francisco. I did a postdoctoral fellowship at St. Jude Children's Research Hospital, where he currently is an associate member and director of the lead discovery informatics at St. Jude's. As far as his development as a researcher, so currently his research is funded by the NIH, but over the years he's had a number of NIH funds in addition to funding from different national and international funding sources. He has been quite committed to a number of the committees at St. Jude's and been a quite prolific member of the academic community. So he has organized much of symposium at different national conferences, has served as a peer review for numerous journals, including high impact journals such as Science, currently has 2 patents, has been involved in a number of clinical trials at St. Jude's, has a publication record that exceeds over 100 publications, has presented at different national and international conferences over 50 of them. So with that introduction, I would like to leave you with our guest speaker, Anang Shelat.
All righty. Thank you, Arthur, for that introduction. And hey, it's a pleasure to meet y'all. Can you guys hear me okay?
Yes, yes.
Yeah, so I want to keep this lecture informal. And so I'll try to keep an eye out for questions, but if I'm not responding, just interrupt me and ask your questions. I mean, these lectures are meant to be useful to y'all, to give you a sense of what the types of research kind of at the cutting edge in terms of from my background, cancer biology and translational research in pediatric cancer. So the topic that I'm going to talk about, so let me share my screen here. Can you guys see my PowerPoint presentation? Does that look all right?
We can see it now, yes.
Yeah.
Okay. So I am going to talk about our work in targeting the DNA damage response. And I'll be honest with you, like when I was a graduate student, I thought that this stuff was kind of boring because the way it was presented, very old school. I mean, you're like repairing DNA, like how exciting is that? Aren't there more exciting sort of topics to think about in terms of cancer biology and the cutting edge of drug discovery? But we are really in the golden age of targeting this pathway for cancer. And the reason why is that DNA repair is really essential to life, and therefore cancer cells have deficiencies in DNA repair on purpose, and those deficiencies can be exploited for therapeutic purposes. And fundamentally, DNA repair is about preserving life because the DNA encodes the information that you use to encode your genes and proteins and such. And so what I have come to appreciate is that in your body, various cells have a different sort of varying levels of tolerance to DNA damage. Some cells in your body, like your blood cells, are really not tolerant of DNA damage. They're going to die quickly as soon as they're exposed. And if you think about people who've been exposed to radiation, what they die from is essentially like blood failure, right? Because their red blood, their blood cells are all dying and creating this sort of huge deficit because those cells are replaceable. And your body's view is, why would I risk mutations in these cells becoming a leukemia when I can just kill those cells off and regenerate more? And then there are other types of cells in your body, like neurons or cardiomyocytes, that are extremely resistant to dying from DNA damage. And so they will tolerate high levels of DNA damage because they are irreplaceable, right? Think about a neuron that you train your entire life to do some task, right? Neurons are generally like terminally differentiated. They don't replace, they don't replicate. You don't want to lose that neuron and what it's doing for your body. And so those cells are sort of trained to tolerate a lot of DNA damage. And so when encountered with things like DNA damage, they use different pathways in order to resolve it. And so what I've come to appreciate is DNA repair is a sort of like plastic process. And cancer cells hijack these repair processes in order to survive, in order to tolerate the types of environments where cancer cells need to survive it. And this is a complicated process that we're still trying to sort out some of the really fundamental questions. And what you're seeing in the background of my slide is really sort of a snapshot of all the different actions that happen when a cell detects DNA damage. And in order to sort of understand the sort of research that we're doing, I first need to give you sort of a foundation on the DNA repair processes that are typically sort of go on in a cell, so you can kind of have some context. So the focus of this lecture is I'm going to focus the first half on really a primer of DNA repair. I teach a class on DNA repair in the cell cycle in the graduate school, and I really enjoy that. So hopefully I can communicate that enthusiasm to y'all and try to convince you why this is interesting and worth knowing. And then I'm going to shift over to try to make the case that we're really entering this sort of golden age for drug discovery. Used to be that DNA damage, like our toolbox, was really just generating damage on DNA. But now that we figured out the signaling cascades, right, we're actually developing molecules to perturb these repair processes, hopefully take advantage of them, especially in the context of cancer. And then I'm going to close out with describing an active research project in the lab that uses this technique called genomic scar assessment to try to find those tumors that might have underlying DNA repair deficits that we could exploit for a therapeutic purpose. So again, please feel free to interrupt me and ask questions. I'm happy to stop and go over any of these details. So let's start off here with a primer on DNA repair. So here are the kind of main points that I want to convey in this section is that your cells are constantly being bombarded by DNA damage. And so they're constantly having to repair their DNA. There needs to be cellular machinery in place to prevent unwanted changes to DNA. This is vital to the propagation of life, right? So DNA repair is about preserving life. As I mentioned before, the tissues in your body have different tolerance for DNA damage. Why? Because some you absolutely cannot lose, right? And some cells are expendable. And so there's this sort of spectrum of how cells tolerate DNA and how they resolve their DNA if they need to fix it. And sometimes they choose options which are not accurate, right? That because they're the fast option, they're the best option, sometimes they're the only option to repair DNA. So they will tolerate mutations because the consequence of not fixing that DNA damage is death or the inability to replicate. DNA repair deficiency can lead to many diseases, including predisposition to cancers. But cancers are often sensitive to inhibition of certain DNA repair pathways, and this is a booming area of interest for drug discovery, and I'm going to highlight some cases. So just this is just really kind of basic stuff, just to jog your memory a little bit. The structure of a DNA monomer. Right, so you have the nitrogenous base, you have the sugar component, you have the phosphate. DNA is replicated 5' to 3' on your DNA strand. The 3' hydroxyl attacks this, triphosphate on the nucleotides. So just to remind you that a nucleoside is the base plus the sugar. A nucleotide is the nucleoside plus a phosphate. You have the purines, adenine and guanine, and you have the pyrimidine, cytosine, thymine, and uracil. Now, in any given cell in your body, right, these are the sort of frequency of DNA damaging events. Now, you have like 3 billion base pairs, so these numbers are actually small, but think about it that in any given cell in your body, there's about 50,000 single-strand breaks happening, right? And multiply that by the trillion cells in your body. There's a lot of DNA damage going on. It's a natural part of existing. And so single strand breaks are the most common type of damage. You also have depurination or depyrimidization, the loss of this base or destruction of this base, which happens at high frequencies. And then you have various other types of DNA damage. And at the bottom of this list, the most infrequent type of damage, thankfully, are interstrand crosslinks or double strand breaks or DNA protein crosslinks. And these are incredibly toxic to a cell. Why? because those types of adducts prevent the cell from replicating. And so thankfully they're at sort of low frequency, but even think about just in a single cell, you're going to encounter this naturally as part of existing and multiply that by the trillion cells in your body. And so DNA repair is constantly going on in the background at a high level. And so just to ask you guys a question here, notice this discrepancy between depurination and deperimerization. Why is there such a discrepancy in terms of this DNA damage? Does anyone want to guess why those two are different? And take a look at the structures of these bases. It's a chemical reactivity question. Anyone have a guess? So depurination happens at a much higher rate than deperimerization. Even if it's wrong, just chime in.
Because if you cleave the chemical bond between the sugar and the nucleotide, then due to the fact that you have like electron density moving around the aromatic rings, that it can like stabilize a negative charge at those positions.
Absolutely right, right? See, these guys are more prone to basically being lost because the nature of their rings, it allows basically, some sort of nucleophilic attack where this base can be released. They're also more chemically reactive and more prone to damage compared to the pyrimidids. And that's why you see this sort of like, tenfold difference in terms of the damage that's created. And it boils back down to chemistry, right? And so this is sort of the theme that you should consider in terms of DNA repair, is that there are all sorts of different ways that your DNA molecules can be damaged. And through time, through evolution, your body has developed, or life has developed, these different types of repair mechanisms to account for the types of chemical damage that's happening to your DNA. And so the general strategy for DNA repairs, first you have some mechanism to repair. So there's some surveillance mechanism. Then you launch the DNA damage response. This is a signaling cascade. This tells the cell to hold on, right? If depending on the nature of the damage, we're going to arrest our cell cycle. If the damage is extensive enough, and this is a type of cell that's replaceable, it's going to commit to cell death or apoptosis, right? But in order to repair the damage, right, you just don't have naked DNA, right? Your DNA is packaged into chromatin, so you actually have to figure out how to remove that chromatin so that you have access for those repair enzymes, and then you have to put everything back, right? So this is actually a complicated process that requires a lot of coordination, and that's called the DNA damage response. So think about the different types of chemistry in terms of the different types of damage that can happen. So free radical damage can create structures like this, where instead of you have a 3' hydroxyl, right, you have a phosphate on the side and a hydroxyl on that side. Or you could have part of the sugar base through radical damage, linked to this phosphate because the base and the different components just blew apart. There's a cancer drug called bleomycin, which induces free radical damage, which creates these types of double strand breaks where you have part of the sugar connected to the phosphate group. And so the chemistry here is you cannot use the normal DNA replication machinery or ligation machinery to just stitch this back together. You actually have to use chemical reactions to clean up the ends of these broken DNA components. There's a drug that we use in the clinic, or a class of drugs called topoisomerase inhibitors, and they trap an enzyme called topoisomerase, which is involved in the relaxing of the torsional stress of the DNA molecule. And those drugs actually trap the protein on to the DNA and create these bulky sort of adducts, which block the replication machinery and sometimes the transcriptional machinery. So this is a protein DNA adduct. You have specialized enzymes that actually clear those proteins away from the DNA to allow you to actually then ligate that DNA back. And there are classes of enzymes called glycosylases that detect damaged bases. And so they basically destroy the damaged base, sometimes based on the chemistry, right? They can have side reactions that create products like this. And so you have to have repair machinery that recognizes this particular chemistry in order to clear that and restore the sort of DNA to a sort of 3' hydroxyl 5' triphosphate so that you can ligate back. So, defective DNA repair is really an interesting concept for tumors because this plot here, this graph here is from the hallmarks of cancer. So people who have studied cancer have basically defined the properties that are present in all cancer cells, like evading growth suppressors, avoiding the immune system. But one of the hallmarks of cancer is genomic instability and mutation. And so why is that? Why do cancers tend to select for environments for genomic instability? Well, first of all, that's part of the reason why you get cancers is you have genomic instability, right? But these unstable genome environments, they basically allow, they accelerate evolution. They allow the cancer cell to adapt to different environments. A cancer cell is a cancer cell because it's doing something that it's not supposed to do. Normally in your body, your cells are under tight control from its neighbors and from external signaling that says, this is when you grow, this is when you don't grow, right? This is what you're supposed to be doing. A cancer cell loses that identity, essentially loses those ability to process those instructions. And so it has to adapt to the environment it's in, especially if it's mobile, metastatic cancer cell. And so genomic instability allows that sort of regulation that your normal cells have. It sort of blows that apart and allows the cancer cell to transcribe genes that it didn't transcribe before, but also to pick up mutations to activate signaling cascades, to basically create essentially new protein structures that allow it to survive. A cancer cell is very selfish. At the end of the day, it's trying to preserve its own existence. And so it's using the mutations in DNA to basically evolve to the different environments it's faced with. And this creates a double-edged sword because that genomic instability is important for creating and sustaining that cancer cell, but it can also be targeted. Too much instability basically makes replication unsustainable and can be used to kill those cancer cells. So I want to briefly step through the sort of main repair processes, because in order to understand what I'm going to talk about later, you kind of have to sort of general sense of the different repair processes. So these are the sort of main families, and not shown in this graph are there several sort of like specialty enzymes that have very specific sort of, it fixes very specific type of damage. And a lot of these are actually sort of like suicide enzymes. So they'll go in there and they'll repair the damage, but they also inactivate themselves because, again, it's a chemical reaction that gets spent. And so one of these is called MGMT, and I'm going to talk about this a little bit later in the lecture. But these are sort of the sort of primary repair pathways, if you will, and the relevant effectors for each of these guys. And so we've actually, in our armament for oncology, we have developed drugs which actually target these various pathways because these pathways are defective in certain types of cancer cells. And in fact, that's maybe one of the reasons why that cancer developed is because they had a deficit in one of these pathways. And so these drugs are used to sort of target those tumor cells that originated from that deficit. And then these are the types of sort of damage that these agents sort of cause, and then the underlying sort of like, predisposition diseases that are tied to defects in these pathways. So let me just walk through these pathways to give you a refresher on what they are. The first one I'm going to talk about is base excision repair. And so this process repairs the most common type of DNA damage, and those are single strand breaks. And single strand breaks can have sort of two flavors. The sort of like very sort of simple version is that, you in one of your strands, you are missing a base or more, but it's only on one strand. And so that other strand, that information from that other strand is available. In other cases, the single strand is not actually broken, but it has an AP site, apurinic or apyrimidinic. So it's lost that nitrogenous base component. And so it needs to be fixed, right? And so in terms of these apurinic sites or pyrimidic sites, AP sites, there's an enzyme called APE1, which recognizes those sites in particular. It recruits polymerase beta, which specializes in this gap filling. And it basically like clears the AP site, sort of like makes it so that it's a single strand break and it has a 5' hydroxyl and three, the ATP can come in and sort of like fill in the gap, and then ligase comes in and connects everything together. For these types of structures, there's an enzyme called PARP1, which recognizes the sort of like single strand gap and recruits machinery, and through a similar process with a DNA polymerase, fills in the gaps and ligases everything together. So one question is that this pathway is really highly active in the mitochondria. So can anyone guess why this pathway is really active in the mitochondria? What type of environment does the mitochondria have in terms of chemistry? What's going on there? Anyone? Mitochondria? Energy production. So remember the electron transport chain. What do you generate in terms of what electron transport chain, electrons moving around? You have a tendency for generating free
radicals.
Exactly. And free radicals, right, can damage things like DNA. There's also reactive oxygen species, right, which are like free radicals, part of the peroxides that you can generate. Again, the way the electron transport chain works. So mitochondria has high levels of ROS going on, and this can damage the mitochondrial DNA. And so these repair processes are really active and important in maintaining the quality of your mitochondria. All right, so the next repair process I'm going to talk about is mismatch repair. And so, we think about sort of the canonical forms of base pairing, right? C to G and A to T and this sort of stuff. But, your nitrogenous bases, those are chemicals, right? Those are molecules. And we know molecules can do things called tautomer shifts, right? They can exist in these multiple tautomeric forms. And these tautomeric forms can cause non-natural base pairing. Right, so here's an example that here are your, ATCG, and here are the rare tautomers that can exist just naturally, right? It's not a mutation. It's just this sort of oscillation between where the protons are on your chemical structure. And if you happen to have this sort of rare form of this tautomer, you can create a mismatch, right? Not a mutation, but just the way the chemistry behaves. And so there are other examples where these mismatches, they can go to a base pair that's directly across or kind of like staggered, and these create mismatches. And this is naturally happening as part of chemistry. And where this is really sort of deleterious is after you've replicated your DNA. So for example, if your polymerase goes and it detects this altered form where it's going to bring this base in where it actually should have been a guanine. But this is what formed the best base pair, and your DNA polymerase was, all right, it went and it actually catalyzed that chemical reaction. And so you have developed surveillance pathways to detect these types of mismatches that happen from tautomer shifts or sort of natural wobbliness in the base pairs. And so the first one is actually built into your DNA polymerases. So your replicating polymerases are polymerases delta and epsilon, and they have a proofreading mechanism. So after they actually catalyze the reaction, they bring in a new nucleotide. It's using the template from that strand. And so the base pairs happen, right? And it catalyzes that reaction. Then it moves on to the next base pair. There's a proofreading domain that looks at that sort of base pair and says, is this actually right? Is the geometry actually correct here? And if it detects that it's off, then it actually causes the polymerase to unwind, excise the bad base, and then it basically like replicates that area again. And so if you introduce a mutation in the proofreading domain, right, a single mutation, because you have two copies of this gene, is tolerated, right? But if you have a mutation in both of your alleles for the, this is polymerase delta in the case, right? Then you get 100% incidence of cancer. You become hypermutagenic. The mutational burden is so high that you're guaranteed to get cancer with just that single mutation, biallelic mutation in this proofreading mechanism. So that's one repair mechanism, but there's another repair mechanism that after the polymerase has sort of moved on, there's a second quality control check, and these are the mismatch repair proteins. And in humans, this is like PMS2, MLH1, MSH2, 3, and 6. And so what this is doing is this is actually creating sort of a clamp that's constantly surveilling DNA, especially post-replication. And it's looking for distortions in the double helix. And when it detects a distortion in that double helix, basically it's going to cut out the newly synthesized DNA, because what it's doing is it's saying, well, the original version is the correct copy. What I just synthesized is where the error is. And so I'm going to cut out the newly synthesized version, and then I'm going to fill it in with a ligase and repair it this way. And so what is interesting about these mismatch repairs is that, they're germline predispositions that cause you to have these defects. And typically you show up with one, like you inherited one bad copy, and then at some point in life, you get a second bad copy. So now you're biallelic. And so you have a deficiency in this repair process. And DNA polymerases tend to be more error prone in regions where you have a bunch of repeats, like a bunch of ATs especially. GCs and ATs, these repeat regions, exist in your DNA. The AT regions are particularly a concern for polymerases because the AT base pair is weaker, right? It only has 2 hydrogen bonds compared to the GCs, which have 3. And your polymerase, like it tends to slip in these regions because you have a bunch of repeats, right? Sometimes it can be confused, which am I supposed to be, replicating at this index or at this index? And so it can get off register. And so this is called stuttering. And so in these regions, they're called microsatellites. People with these deficiencies in the mismatch repair tend to have lots of mutations, particularly in these regions. And it actually becomes a diagnostic. And what we've been able to do now is look in the genome specifically at these regions of ATs and GCs, these repeat regions, and actually classify them. And based on the types of mutations that you see there, you can actually infer whether you have a mismatch repair and what type, right? Whether it's coming from the polymerase or whether it's coming from these mismatch repair genes. And to add another level of complexity to it is that these types of scars, these genomic scars, these patterns of mutations tend to be tissue type dependent. So in a patient with mismatch repair, it could be that in the colon, you see these types of mutations in these microsatellites, but in breast tissue, you don't see it. And so there's a tissue type dependence to these genomic scars, these patterns of mutations that also sort of complicates it. But it's kind of cool that you can look at the patterns of mutations and infer what the repair deficit might be. And in particular case, what particular enzyme might be defective or repair pathway might be defective. If you know that information, then maybe you can target it with a drug. All right, the next repair process I want to talk about is called translesion. And what's interesting about this one is that it's actually not so much of a repair process. Sorry, am I looking at, hold on. My screen is blocking me, so I can't actually see. Sorry, I jumped ahead. This is nucleotide excision repair. So what this repair process is really tied to thinking of when your DNA damage, the helix is disrupted by damage between bases, like from UV radiation causes these things called thymine dimers. So on a single strand, the adjacent bases are basically chemically reactive, right? Or bulky adducts that are sort of like, usually with like small molecules. So for example, when you cook meat, you generate compounds called polyaromatic hydrocarbons, which are chemically reactive, and they can create these bulky adducts on your DNA. And so you have NER is the process that deals with the repair of these. And this repair process can kind of work 2 ways. You have a global surveillance sort of mechanism. Again, these protein complexes that are constantly surveilling, but you actually have a mechanism that's tied to polymerases, so transcription. So you're transcribing along, right, with your RNA polymerase, and all of a sudden you detect one of these bulky adducts, you can activate these NER process to come in here and actually fix the DNA. And so you're actually leveraging a transcription factor that initiates the signaling cascade. again, the thought process here is that with this bulky adduct, I'm not trying to fix this single base. I'm just going to cut out this section, right? And create this sort of like gap. And then I'm going to use the other strand and as my template, recruit a polymerase, which is going to fill in that, and then ligases, which tie everything together, and your repair is fixed this way. The next repair process, translesion synthesis, isn't actually a repair process, it's a bypass. So you have evolved a way to basically ignore DNA damage. And where this comes into play is usually when you're replicating your DNA, and the replication machinery, it encounters one of these damaged sections of DNA. Typically, like it's a cross-link, so it really can't unwind anymore. And so it just like physically stalls, right? Like an engine that's stuck. And so your cell has a choice, is to either, well, commit cell death because I don't want to tolerate this DNA damage, wait for other repair machinery to maybe like try to fix this. That's the first sort of bet. But if there's no way I can fix this damage, right? It's a chemistry that my enzymes haven't sort of evolved to deal with, then I recruit these translesion process, which allows me to basically skip over, right? So it's sort of like takes the replication machinery, allows it to basically like break apart and then restore back after the DNA damage. And so the damage is still there, but the cell has basically now allowed to continue replication. Why would you have this process? Why wouldn't you want to fix this? Why would you tolerate mutations like this, right? Because ultimately this is going to create, some sort of distortion, some sort of mutation, because you have this damage in your DNA that your cell basically like replicated past, right? It's because this cell wants to survive, right? It's made a decision that it is more important for me to replicate and get on with my cell cycle because I serve an important purpose than to stop and basically freeze at this point and risk cell death. And cancer cells in particular leverage this pathway to deal with the genomic instability that they normally have, but to also become resistant to the type of DNA damage that we use in chemotherapy. And so this pathway is really not well understood. We have some sense of it, but there are various components, and we're right at the cusp of being able to drug it, but it could be a very attractive target for the next generation of, targeting DNA damage defects in cancer cells. All right. A lot of those like BER, NER mismatch repair, those are basically, repairing one single strand and using the other strand as a repair template process, right? Now we're going to talk about double strand breaks, which are the more toxic type of DNA damage to cancer cells. And in terms of double strand breaks, you have two repair processes: homologous recombination, right? Which is relatively error-free, because what this uses is the second copy of your DNA that you have at certain points in your cell cycle. So when cells are replicating, right? When you go through the cell cycle, at some point you have a second copy of your DNA right before you basically replicate into two daughter cells. That second copy is essentially a backup, right? Like a backup of your hard drive. And so homologous recombination uses the information in that second copy to repair the damage in the first copy. Non-homologous end joining is basically the repair processes that happens at every other point in the cell cycle when you don't have that second copy, right? You don't have that second copy, so what do you do? Well, you figure out ways to basically put these double strand, these two pieces of DNA that are broken apart to fix them back together. The thing to think about is that these are not like single processes. They exist on a spectrum. So both homologous recombination and non-homologous end joining, they can be very error-free, or they can be very error-prone. And that's not typically the way you're taught it in sort of like normal cancer biology. You're taught that homologous recombination is error-free and non-homologous end joining causes a lot of mutations, but non-homologous end joining is the primary repair pathway for most of your cells, because in your body right now, most of your cells are not actually dividing, right? A lot of them are just terminally differentiated. And so if they're not dividing, well, there's never that second copy. It's never going to be an S phase. You never can take advantage of homologous recombination. So you leverage this process. And so we're going to walk through these repair processes because understanding them is really key to how we're going to target them later in the lecture. So the first repair process I'm going to talk about is homologous recombination. Again, there are different names for this: strand displacement, holiday junctions. These are sort of the terminology. You know, basically the idea is that when you have your double strand break, you can take one strand on one of the break, sort of one side of this double strand break, and it invades that second copy and uses the information from that second copy to actually as a template. And then once you get enough information from that second copy, eventually now you're going to be able to base pair back to that other end, and then you can fill in the gaps. But rather than explaining in this way, I think I'll just show you this video, which, well, got to switch this over. Okay, which I think does a better job of explaining it. Have it queued up here. Here we go. And just, you guys can hear the voiceover? Very.
No, we also can't see the video.
Yeah, we don't see the video.
You can't see the video. Okay, hold on now. Let's see. What are we doing here? How about here? Can you see the video now?
Not yet.
Not yet.
Okay,
Now.
And could you hear the voice over?
No sound, no.
No. Okay. Well, I'm gonna give you the voiceover.
Okay.
But you can see the video, right?
Yes. Yeah.
Okay. So in this particular repair process, what's happening is, so again, you have your two copies. Here's your double strand break, right? And so you are recruiting this repair machinery tied to homologous recombination. And what it's going to do is it's actually going to excise one of the strands. So you have these single strands, right? And now you can use these single strands to invade that second copy. So here's to talk about your single strand. And there's protein machinery that detects the corresponding region, right? The homologous region on that other copy of DNA, right? Allows that single strand to invade in and base pair. And now you have information from that second strand that you could use to basically replicate from. And so this creates a replication bubble. You're going to go through here. It's called the D loop. This is a polymerase that gets recruited. Boom, boom, boom. I'm using that second copy as my template. I'm creating a double helix, just normal replication, right? And so I'm going to go to a certain distance, right? Where now I'm actually copying a part of the strand that exists in that broken end, right? And so now I actually have some overlap from the damaged copy of my DNA. And that's critical because now my single strand can leave that second copy and it can go back to the first copy and it'll base pair correctly because I have now enough information for it to basically find the homologous region that it's supposed to in that broken part of the DNA. And so now I know what to do, right? Because I have my template strand here. I can recruit my polymerase, boom, 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 fill in the gaps here, ligate, and everything is cool. So here's that polymerase going, using that information that it just copied from that single strand, filling in all the gaps, ligase goes, and back to normal, right? And in this particular repair process, right, I preserved all the information in C because I was able to use that backup copy and I didn't lose anything. And that's why homologous recombination is considered a high fidelity process because it's using that backup copy. So back to my screen here. Can you see my PowerPoint now?
Not yet.
yet. Uh,
We're looking at your YouTube.
okay, how about now?
Yes.
Okay. So that's homologous recombination, but like these repair processes, there's backup, there's fallback positions, right? And so typically the way that this HR would go is that you're doing this strand invasion to that second copy, and there are various ways that you can use the information from the second copy to basically restore what the missing information was where you had the break. So again, why are we doing all this sort of stuff? You can't assume that the break was clean, right? So you basically have two components of your chromosome here. And so rather than just piecing them back together, right? I'm going to assume that I've lost some information. And that's why I'm going to resect back and have these single strands. And I'm going to look at that second copy. And I'm going to basically use that as a template. And I'm going to create, again, a big chunk of overlap. And I'm going to use that to go back to the first copy. That ensures that you didn't lose anything. This double strand break could have happened that you just blew apart a whole gene, right? Lost a whole chunk of DNA. It's just not a clean break here. And so this process allows it guarantees that you basically don't lose this information. However, this process can break down because like anything in biology, you need all the protein machinery to work and sometimes stuff happens, right? And so there's a sort of a backup for HR when these are not available, these somehow fail. For example, what if like you had this break here, but this part of the chromosome, it just like floated away or just got degraded, or it's not accessible at the moment. Well, you go and you're doing your strand invasion, but there's nowhere for it to go, right? And so you've gone and you've used a second copy and you've done your replication, but then it really there's no way for it to go back to that broken copy because it's lost that track of that, the other half of your broken end. Well, the cell could just stop there and die, or it could say, wait a second, we have to figure out some way to survive here. And so it could basically say, well, I'm just going to keep on just copying whatever was on this other copy and keep on going, in which case you've basically potentially can lose information because it really doesn't know what's going on here. In some cases, it just stops completely and calls it a day. It breaks this process of connecting back to this. In some cases, it might find the wrong DNA for the single strand to basically find back. So it basically finds another chromosome where it had enough homology. And so it now connects, creates a gene fusion event. This process called break-induced replication tends to happen when you have a lot of double strand stress, like for example, with genotoxic chemotherapy, but also when you have defective homologous recombination. And so this again will create types of mutational patterns that you can detect. But anyway, the whole point of this process, why did this cell have this process, is that if this somehow breaks down because there's some defect in a downstream process, or you were just unlucky, right? Rather than being stuck where you have your two copies and one is inserted in the other and you don't know what to do there, there's some fail-safe mechanism that allows you to resolve this process to free up these two copies of your DNA so that you can go on with cell division. And a cell makes that choice whether it's willing to tolerate that mutation or not. All right. And so now we're going to talk about the second repair process, homologous recombination, and I'll have another video for that as well. So we're talking about here. And so you see the screen or not?
No.
No. All right. Switch over. How about now?
It's like loading up. Yeah. But now.
All right, so in this particular repair process, you don't have that second copy to leverage, right? So how is your cell supposed to figure this out, right? So here, you only have this one copy of your DNA. you're going to have this double strand, very dramatic double strand break. And so what the cell does is it needs sort of like clean ends. And so this repair process is evolved to deal with all sorts of complicated damage. In the case where you just have a clean break, it can just slap it together and you don't have any mutations, it works fine. But in situations where you just have gnarly ends, right? The way it's kind of evolved to fix it is it basically just cuts back. It excises the ends of this DNA until you have blunt ends, literally blunt ends that you can slap back together, almost like you know putting two pieces of wood or whatever, just slap them together so it has a clean surface, and then you go on with your business, right? But the issue with that is that you've basically excised, you removed part of your DNA sequence. You've lost information here. So this is illustrating here that previously what you had was you had the C gene, right? But the C gene was lost because in that excision process, you've cut back in order to create those blunt ends. And so non-homologous end joining can be mutagenic, right? But on the other hand, if you just have a very simple DNA strand break, it'll just slap them back together if you have a clean break, and it doesn't have to do this excision process. And so you won't have that mutagenic process. And so that is why a repair process that exists for hold on here. Back to my PowerPoint. You can see my PowerPoint now?
Starting to load up. We see it now.
Okay. Jump ahead here. Non-homologous end joining, the reason why it's there is that it can happen at any point in the cell cycle. It does not require that second copy. It can potentially be error-prone or mutagenic. If the broken ends can be directly joined, it's extremely fast. And this is one of the sort of like features of it, is it doesn't require recruiting all this complicated machinery that allows you to resect back only a single strand and then have that single strand invade and figure out like where on that other second copy of your DNA it's the homologous region. Is very efficient in this process. It's meant to really be fast. And in cells that really need to quickly repair the DNA, this is a very important process for its survival. But if the DNA damage is more complicated, this repair process basically is evolved to deal with all sorts of possibilities. So it'll literally just shave back the DNA. It'll risk losing this information, and then it slaps the two pieces back together. And now you have repaired that double strand break. You might have lost information. NHEJ is actually used for your immune system, right? It's used in this process called the VDG recombination in your B cells and T cells. This generates purposeful mutation to generate the sort of diversity in your T cell receptors, for example, so that you can detect foreign pathogens. So you have a purposeful immunogenic process in your body to evolve different types of receptors to detect different types of foreign proteins and components. And so in that respect, it's actually a very valuable component of you being able to survive. And NHEJ is actually one of the principal techniques used to generate CRISPR knockouts. So this is how this works. In terms of a CRISPR knockout, what happens is you're using this Cas9 protein and giving it a guide RNA. The guide RNA directs the Cas9 protein to a specific sequence, the sequence that you want to alter, and it generates a double strand break. And so if the double strand break is put back together without any issue, that's fine, but the Cas9 and the guide are still there. So it's like a catalytic cycle. It's going to go and generate that break. And it could be repaired and it fixed, but as soon as that break gets incorrectly repaired with non-homologous end joining, right, then the sequence is actually changed so the guide no longer binds. And so how do you generate a CRISPR knockout you don't actually cut out the gene. What you're doing is you're generating like an insertion or deletion or a frame shift essentially that screws up the sequence right after that double strand break. So generate a double strand break, use the mutagenic capability of non-homologous end joining to screw up the DNA to cause a frame shift. And now that gene is effectively knocked out. It's effectively disabled because the sequence essentially is a nonsense sequence. So you're either not going to transcribe that gene, or certainly that's not going to translate into a viable protein. So that's when we talk about CRISPR knockout, that's essentially what you're doing. You're not actually excising the whole gene. You're introducing this frame shift. But you can also leverage CRISPR, right? So you can generate a double strand break. If you feed the cell an actual template, right, with the modification that you want, then your homologous recombination machinery, instead of finding that second copy, it could actually find your template and use the information from your template to repair the process. And in that way, you can actually insert a gene or create a base edit. And these two repair processes, again, they're happening in your cell. You need replicating cells to have HR. But one of the sort of techniques that we've sort of developed is to be able, not we, but the field is to actually use inhibitors of this process so that you are biased more towards this process to increase the efficiency of your homologous recombination and essentially your editing. And in fact, now they combine with other inhibitors. So this pol theta is involved in the bypass pathways, that if this is not available and this is slow, there's some bypass pathways that you can also inhibit, again, to bias towards HR to increase the editing efficiency. So this is a nice use case of using inhibitors of this pathway for genomic editing. And so finally, I just want to talk about when the main repair processes like homologous recombination or HR are unavailable, right? You have backup processes. In terms of HR, we had a backup process called BIR. But if again, depending on where you are on this pathway, if one of these downstream sort of components is broken, then you get shuttled over to these bypasses. And two examples of the bypasses are called Alt-EJ and single-strand annealing. And these essentially, they cause an incorrect templating. So again, they're in this process where you're leveraging information from another component of the DNA, but it's the wrong component in this case. And so you're generating mutations this way, even though you're repairing the DNA, because you're using incorrect information. And this will be important in later in the lecture. And so just to close this out, defects in these two repair processes, homologous recombination, non-homologous end joining, they lead to just massive genomic instability. And if you look at your chromosomes, you have all sorts of deformations in your chromatins. You have gaps, you have breaks, you have fusion events. And so this is just from cells where you've taken one of the repair machinery out that's involved in non-homologous end joining. And you could see like, this is the instability that you see. And really, when we talk about studying cancer cells in the lab, this is what they look like. Like their genomes do not look anything normal, because cancer cells, especially ones that are perpetuated in cell culture, they have all sorts of chromosomal anomalies. So when you're studying cancer cells, just know in the background that you're really studying an entity that is very different from normal human cells. And in particular, the ones in cell culture look radically different. All right, I want to close this section out by talking about the actual signaling cascade that initiates the repair process. So we talked about the actual enzymes involved in the repair and how you detect DNA repair, but there's actually a signaling cascade, especially with double strand breaks, which tells the cell to basically how to respond, right? Cell cycle arrest, apoptosis, to transcribe new genes for DNA repair, right? To upregulate the repair machinery. And so one of the early responders to a double strand break is something called the mRN complex, composed of the proteins MRE11, RAD50, and NBS1. And this complex senses double strand damage. It activates a kinase called ATM, and it causes this signal transduction cascade, which has multiple sort of downstream targets, including P53, but also sort of the HR machinery. And so ATM serves as an early critical mediator of the cellular response to double-strand damage. It phosphorylates many substrates, one of them being gamma H2X. And gamma H2X acts as a foundation to assemble the DNA repair machinery. What is this protein gamma H2X? Well, it is a histone component. So H2A and H2B, H3 and H4 are the sort of, and H1 are sort of the fundamental components of your chromatin structure. There's a variant of H2A called H2AX, which has a unique C terminus. This C terminus can be phosphorylated by ATM kinase. And this literally acts sort of like as a wildfire that spreads across the chromatin, right? When a double strand break is detected. And so the analogy that comes to mind is that, I don't know if you guys remember the Lord of the Rings. Do you guys know what Lord of the Rings is, or are you guys too young for that?
They know.
They know. Okay, so the beacons of Amandine. This is when Gondor was under attack, right? I think it's Pippin, right? He lights this sort of like fire, and then that point of that fire is that there are watchtowers that are on the mountain ranges, right? And they look at the fire, and then they light their fire, and then some other watchtower lights their fire, and so it creates this signal transduction of these fires, and eventually it goes to the kingdom of Rohan, right? And Rohan sees that fire and they know like Gondor calls for aid. And so we got to go to Gondor, right? This is exactly what H2X, I mean, exactly. This analogy is perfect, right? So H2X is basically lighting this fire in the cell and saying there is double strand damage that needs to be repaired. And it actually points the way because it emanates in both directions from that double strand break. Kind of a cool sort of process that goes in the background. So gamma H2X is one of the markers we use in the lab to detect these double strand break deficits. And so in a cell that, for example, has a deficit in NHEJ, you have these persistent gamma H2X foci, right, telling you that there's just these double strand breaks that haven't been repaired. Why? Because your repair process is broken. The damage is detected, but the downstream events are broken. And the repair machinery hasn't been recruited to that place in order to fix the repair. So you have these persistent double strand breaks. And in our lab, we've used this as a therapeutic. So we've been studying inhibitors of ATM, right, which I just talked about. And this is in brain tumors called pediatric high-grade glioma. So we're looking at two cell lines and we're measuring gamma HX when exposed to radiation. And so, you know, absent this ATM inhibitor, as you increase the intensity of the radiation, you see an increase in gamma H2X. And so there's just two different cell lines, right? So the early effect, right, within right immediately when you generate the double strand break with an ATM inhibitor is that you shut down gamma H2X signaling, right? So that's not saying that the damage isn't happening. It's a radiation. The damage is happening. The cell just doesn't know it's happening, right? And so that damage goes unrepaired. But if you look at later time points, what you see is that the cells that were treated with the drug, they have enhanced DNA damage. Why? Is it because when you trick the cell into not thinking that there's DNA damage, well, it continued to go on with its cell cycle. So it actually exacerbated the damage. It actually became more genomically unstable because you inhibited that repair process. And this is an assay called a comet tail, which is shown right here. A comet tail is like it shows you like physically how your DNA is looking like. And if you have normal DNA, this is how you run this in a electrochemical gradient. And so your normal DNA creates a nice spot, but when you have sort of like double strand break, damage or single strand damage, you create these tails. That's why it's called the comet tail assay. And when you add the ATM inhibitor, you have enhanced physical damage to your DNA. And the consequence of this is that you can create things called micronuclei, which are basically bits of your chromosome that because of the damage, they weren't replicated properly and they break apart. And you can actually detect this. This is in your cell, you're highlighting the nucleus of the cell, and there's these extranuclear components caused by the genomic instability. So with an ATM inhibitor combined with radiation, you tip this cancer cell over into extreme genomic instability. They already have genomic instability, but now you've exacerbated it, and now the genomic instability is too much for the cancer cell to handle. It's not able to replicate, and effectively it either dies or just senesces because it's no longer able to replicate. And so this work here that was published in 2022 is actually the foundation of a clinical trial that's running at St. Jude right now. And our role in this was to evaluate the drug and show that it was effective in this very deadly tumor type that I'm going to talk about in the end here. And so that's sort of the foundation. I spent a lot of time on trying to talk about these processes because in order to understand the next steps, you kind of have to understand the repair processes. But why I'm excited about this, why we are studying this in the lab is that they're just like new molecules coming out every day that are targeting different parts of these components. And it's fun to tease out where these might be useful for us in cancer, but in our particular case, in pediatric cancer. So these are examples of molecules, right? This one is targeting translesion. This is targeting, you don't just have to target enzyme, kinases. Now we can target the ubiquitin machinery because ubiquitin signaling actually plays a very important role in DNA repair. And inhibitors are emerging in that space. But let me give you a very recent example. We are working on molecules in this space, and it's from a close collaborator, the Binger lab at Yale. One of the drugs we use quite frequently in pediatric cancers is temozolomide. In adult GBM, so brain tumors, one of the deadliest brain tumors that you can have in adult. This is the only approved agent, temozolomide. And so what is temozolomide doing? It looks like it's a nitrogenous base, but in fact what it is it's actually in solution. It breaks apart into this methyl diazonium ion. Essentially, it's an alkyl group donor, right? Highly reactive, chemically reactive. And what this group does is that it alkylates your DNA. And so it alkylates at various portions, right? These portions, these types of damage here are easily resolved by the BER mechanism that we talked about previously. But this type of damage is actually quite toxic to the cell because it causes improper base pairing. There's a specialized enzyme called MGMT that was evolved to deal with just this particular lesion because it is so toxic and deleterious to the cell, right? So its one job, it acts as a suicide enzyme. It has a reactive sulfur group, and it basically does a SN2-like displacement on this alkyl group and liberates back the oxygen, the unalkylated oxygen. In the process, it basically chemically inactivates itself. So you have essentially a reserve of MGMT enzyme in your body to deal with this type of damage. And typically you're not under high sort of like chemical load and you have enough enzyme to sort of deal with it. When you give temozolomide, you can saturate this process and then start generating the cytotoxicity. And so what we use this drug is in tumors that are MGMT deficient. So some tumors have silenced this gene. And it's unclear whether it's silencing this gene or silencing a section of DNA that has this gene. But some tumors naturally have this silence, so they're defective. They're deficient in this enzyme. And so they cannot tolerate this type of damage. So where actually is the cytotoxicity coming from? So in tumors that are MGMT deficient, temozolomide is effective. It actually doesn't come from the alkylation event. So just to work through this process, right? In your DNA, when you expose it to temozolomide, you generate this 0,6-methylguanine. If you have MGMT present, then it repairs it, you're fine, right? But in an MGMT deficient cell, what happens is that the MMR process, which is surveilling the DNA, it looks at this and says, wait a second, this is a mismatch because the O-methylguanine creates an improper base pair. And so the cell is like, okay, well, what is the sort of, what is the thing to repair? When it detects this sort of mismatch, it's going to excise one of the strands. And so if it excises the T component here, right, then it's going to come back and you still have the O-methylguanine, right? So it's going to actually repair this back with another T. And then the mismatch repair is going to say, wait, this is a mismatch. And so it's going to excise the section with the T and it's going to fill it in and you're going to go back a T. So you actually generate a cycling event, we call feudal cycling. Mismatch repair gets confused with this type of abnormal base pair. It doesn't know how to fix it. And so it basically burns the energy, the ATP of the cell, constantly repairing this. Eventually, the mismatch repair process creates double-strand breaks, and that is what's cytotoxic when you have MGMT deficiency. It's not actually the alkylation event. Your cells can tolerate those cancer cells can tolerate those alkylation events. But this mismatch repair process basically creates a runaway cascade, which ultimately leads to double-strand breaks, which is cytotoxic to that cancer cell. So what's interesting is cancer cells have evolved ways where they disable the MMR process. And now what happens is that you basically fix this by using the T as the correct base pair and you put in the A. And so you tolerate mutations, but cancer cells are fine with mutations at this level. And so this is one of the ways that this drug becomes ineffective, right? And so this just sort of illustrates this in cancer cell lines. This drug does nothing when you have MGMT proficiency. So the black line and the blue line here. But if you have MGMT deficiency, right, and a intact MMR process, it becomes cytotoxic. So this is survival here. You're basically losing these cells. But if your cancer cell then loses MMR, you actually restore, you rescue this cell from the cytotoxicity of temozolomide. MMR deficiency is actually one of these cancer predispositions, and it's very exciting that we've developed therapies to actually target these tumors. One of them is immunotherapy, because having this mismatch repair I talked about earlier generates really high frequencies of mutations. Well, those mutations create antigens that your immune system can sort of key into. And if you can sort of boost the immune response, you can actually target those tumors that have MMR deficiency. MMR also has this microsatellite instability that we talked about, and there are drugs that really are important for replicating DNA in these microsatellite regions. And so if you inhibit those, the MMR deficient cells are really sensitive to this type of inhibition. But one of the sort of examples of leveraging chemistry that I wanted to bring up in this case is an analog of temozolomide that was reported in 2022. And so what the team at Yale sort of looked at is like, okay, this is the normal mechanism that we have in terms of temozolomide. And we know that it becomes resistant because in the MGMT deficient background, you need this MMR process to generate the cytotoxicity. So what if we chemically modify temozolomide here to this compound called KL50? And all we did is added another carbon and a fluorine group, right? So this is what this molecule decomposes to this. So instead of having the methyl here, it has the ethyl fluoro group, right? What is the consequence of that? Well, this is going to alkylate on your guanines before. And now you have this interesting chemistry possibility because you have a really weak leaving group, right? Fluorines are not awesome in terms of SN2 displacement, but you do have a lone pair from this nitrogen, right? And if you wait long enough, a chemical reaction can happen and you can cyclize, right? So this repair, it takes time. And when you have MGMT proficient cells, you repair this before the chemical reaction can happen. But if you're deficient, you have these persistent alkylation events. The chemistry can happen. You generate this adduct. And now, look, this is like a manic base, right? These guanines are going to sit next to a cytosine. And the cytosine has this nice lone pair, right? Again, it's not highly chemically reactive, but you actually have a reactive group here. You can now generate an intrastrand crosslink, right? And so what you've been able to do through chemistry, this is just to show you that in these cells, all you need is the MGMT deficiency. Now you become independent of MMR in generating cytotoxicity. And this is showing in vivo that in these cancer cells where you have MMR deficiency, which is typical for temozolomide resistance, you have a very weak response to temozolomide, but you have a very strong response to this KL50 compound. What you've done is you've used chemistry and sort of like that organic SN2 leaving group and the sort of differences in reactivity to generate the sort of context-dependent crosslink. Only in MGMT deficient cells are you going to have this crosslink. And what's cool is if you use chlorine instead of fluorine, it doesn't work because chlorine is too reactive, right? What you need is this time delay to take advantage of the fact that in an MGMT deficient background, you have this persistence. This guy can last long enough in order to cyclize and react with the intrastrand cytosine. Very fun for us to think about. But in our lab, this is work from our lab. So we've been looking at these two compounds. And so first we wanted to ask the question, where is MGMT deficiency in this case, brain tumors? And you can see that in some subtypes of brain tumors, it's actually quite high. 50% of those tumors have MGMT deficiency. And these are two cell models, one of them proficient and one of them deficient. And so you can see with this temozolomide that you have sensitivity in the deficient line, but not the proficient line. And we actually developed a combination strategy by combining temozolomide with an ATM inhibitor to get an even more potent effect. And that's that blue line there. WSD is an ATM inhibitor. And so this is an example of us sort of like looking at this. This drug was developed for the adult space, but we use it in peds. We found the right tumor background. We can have this combination that is highly selective for MGMT deficient cells. And I'll talk about this mechanistically, but we're now pushing this in vivo, and we've found a way to actually deliver these drugs. There's a sequencing. If you put them together, a temozolomide and ATM, it's toxic, but if you stagger them, you take advantage again of this kinetic difference that in MGMT deficient lines, double strand breaks take time to evolve. Then you can actually have a tolerated drug combination strategy to maximize the effect of this drug. And so finally, to sort of wrap up, I know I'm running a little bit late, talk about genomic scars. This is my project team. But we talked about how cancer cells sometimes disable parts of their machinery in order to create genomic instability. And in adult cancers, there's actually ovarian and breast cancers, a subset have mutations in BRCA1 and 2. This is a germline predisposition. And so that disabling of the HR machinery creates genomic instability, but in those cancer cells, it creates a dependence on an enzyme called PARP1. And PARP1 inhibitors have been developed, and they actually show really dramatic responses in certain backgrounds. And so this is a survival plot in patients showing that if you give them one drug, right, as a maintenance therapy, you can actually cure these patients for multiple years effectively. It's sort of surprising that one drug can have this effect in the right patient population. And now the drug industry has developed drugs that actually can go into the brain and are CNS penetrant. And so I mentioned before how in the repair process, if you have some sort of defect in this downstream process, you can actually have these bypass processes which are mutagenic. And so there are ways that you can look at your DNA based on patterns of mutations at the chromosomal level, at individual base pairs, looking at patterns of insertions or deletions, again, looking at chromosomal level in a different level. You basically are looking for recurring patterns in the types of mutations that are present. And the field has identified certain patterns that are associated with deficits in HR. And so the goal of our project was to basically see, like, are these mutational patterns present in our pediatric case? In fact, these mutational patterns can be better than actually looking at sequencing data. And so in this study here, what they did is they took a bunch of patients and they tried to look for actual mutations in BRCA1 and 2. But then you looked at mutational patterns, and they found that a larger number of patients had the mutational patterns consistent with an HR deficit, and this was actually a better predictor of drug response. So what was happening is that there are different ways you can alter this pathway. If you just focus on BRCA1 and 2, you miss patients that have mutations that are in other parts of the pathway that could be sensitive to PARP inhibitor. And so we applied these mutational patterns to pediatric high-grade glioma. It's the deadliest, one of the deadliest tumor types. It's the leading cause of brain tumor deaths. The deal with this tumor is that it's embedded deep inside the brain. And so surgical resection is difficult. And the only sort of tools we have, it's chemo-resistant, is radiation. And so what we tried to do in this study is we amassed a large collection of whole genome sequencing data and RNA-seq data, and we did an analysis of those genes to try to look at these different mutational scars. And again, I'm hurrying you here for the sake of time. We did not detect any evidence of BRCA1 and 2. But as you flip through these different genomic scars, on the left side here is what you find in BRCA patients, right? So here's a cohort where you have, in the red and purple, patients that have BRCA1 and 2 deficiency. In the green, those are wild type. And these mutational scars, the ones that have the mutations in BRCA1 and 2, have an elevated signature on this particular genomic scar. Well, there's a subset of our high-grade patients that also have the elevated signature. For this one, SBS3, this black is the signature that's present in the BRCA1 2 patients. And we see this in high-grade, even though we don't have BRCA1 and 2 mutations. In this particular case, this insertion deletion signature, we see that in BRCA, but not in their high grades. And in this one, in the structural variants, we see it in both cases. So putting this all together, just skipping ahead, we integrated all of these genomic scars and we created this sort of score for our patient population. And we were able to actually look at cancer models that were derived from these patients in our population. And so the first thing that we wanted to do is this: does this score actually predict for sensitivity to PARP inhibitors? Because they don't have the classic mutation, but maybe have the scars that are telling us that they actually have an HR deficit. And so as a control, we engineered BRCA knockout in the cells and showed that they respond to PARP inhibitor. This is a microscopy experiment. The more blue you're seeing, the more cells that are present. And you can see in the wild type cells, they don't respond to a PARP inhibitor. In the BRCA2 knockouts, they respond to a PARP inhibitor. And then putting this all together, we took models that sort of span the spectrum, right? You have more genomic scars up here and less down here. The cells down here they actually don't respond to a PARP inhibitor. So this is looking at just two different time points. Again, you see more blue happening because the cells, they don't care about your PARP inhibitor. But the ones here that have more genomic scars consistent with HR, they actually are responding to the PARP inhibitor. And so why this is important is that nobody thought in pediatric high grade, one of the deadliest diseases out there, that there was HR deficit enough that you could actually have sensitivity to PARP inhibitors. They didn't have any BRCA1 and 2 mutations, right? And so using these genomic scars, we actually inferred that they do have the defects in this pathway, but they're not your canonical versions. They're hitting something else in the pathway. And we are able to show in models that we could sort of associate with patients that were on high or low end of the spectrum, that it actually predicts for sensitivity to PARP inhibitors. And so this is exciting for us. We're trying to develop a biomarker guided clinical trial in this patient population, which again, there are not a lot of good options for. Most of the patients, 90% of the patients that have pediatric high-grade glioma will die within the first 2 years. And so this is exciting. A subset of the patients might respond well to this type of therapy, but we have to find the right subset. So in that, I'll close. And I'm sorry I run a little bit over, but if there are any questions, I'm happy to answer them.
All right, thank you. Any quick questions? Let's give a round of applause for our speakers.
Hey, thank you for your time. And if after the fact you have any questions about this lecture or in general about stuff that's going on here, my email address is anangshalat@stjude.org. Happy to talk with any of you. Guys, have a good day.
All right, thank you. You too. Nice, take care.
Okay, bye.